All right, let me start the recording. Recording has started. All right. So chapter, our topic is going to be female gametophyte. So this is where we'll understand everything in terms of the female gametophyte. That's your gynoecium, right? So that will be the objective of the class, to understand the female reproductive organ and megasporogenesis. What is your megasporogenesis? These are the two things we'll try to understand over here. And after understanding these two things, we'll be solving roughly around more than dozens of questions we'll try to solve, right? So 12 से 15 क्वेश्चन हम लोग solve करेंगे. We'll try to solve like 12 to 15 questions just to have a better understanding of the thing. Okay. Now, when we discuss a female reproductive organ, we are basically discussing over here the gynoecium, which can be also called as my carpel, which can be also called as your carpel. Now, when we discuss about the carpels, you should know one thing: the what a carpel is going to have. So carpel will be having these three parts, which you can very clearly see over here. It will have the stigma, that is the landing platform for the pollen grain. So pollen grain are actually going to come and sit on the stigma, right? So when the pollen grain are coming along with the wind or along with the insect, they are basically going to sit on the stigma, right? Then you have a style. Style is the area through which you will be finding the pollen grain. The pollen tube will reach in the depth of the ovary. Okay, and then the lower part over here, this lower part is going to be called as my ovary. This is going to be the ovary. Please remember this thing very clearly, that in the flower, in future, the ovary turns into the fruit. Ovary is the one which will ultimately turn into the fruit, right? So that is something you should know. Something we'll discuss that later in detail. And inside the ovary, you will have a presence of the ovule. So this should be very much clear to you, that inside the ovary, you will have a presence of the ovule, right? Stigma is a landing site. It is a landing site for your pollen grain, right? Style is the long neck-like region. Style is the area through which you will find the pollen tube can reach up to the ovule. Okay. So that is something you have to know. So this is what you will get to see in a carpel. तो ये चीजें आपको carpel में देखने के लिए मिलती है. This is what you will get to find in the carpel. So you will have a stigma, you have a style, and you have a ovule. You have ovary, and within the ovary you will have the ovule. So this thing should be very clear to you, that ovary in future will turn into a fruit. Ovary is always going to turn into the fruit, and your ovule in future will turn into a seed. After fertilization, ovule ultimately turns into the seed, right? So that is one thing you should be very much clear with. Okay. Now there's one thing you have seen when we were studying about the male reproductive part. When we were actually understanding something in terms of the male reproductive system, we were discussing anther. Everyone knows that we were actually discussing over there the anther part. I think just wait a minute. We were discussing something in terms of the anther over there, right? Now there's one thing you have to understand here is. अच्छा, by the way, I hope सबको summer break का form मिला है, right? I think Mohit sir is here, so he's going to like.
सर मुझे ना सिर्फ 2 मिनट लगेंगे बच्चों को बताने के लिए. थैंक यू सर. बच्चों आप लोगों को ना ग्रुप पर एक गूगल फॉर्म मिल जाएगा आपके समर ब्रेक के रिगार्डिंग. हम बच्चों का समर ब्रेक जानना चाह रहे हैं, कब से समर ब्रेक स्टार्ट हो रहे हैं और कब आप लोगों के समर ब्रेक खत्म हो रहे हैं. इन समर ब्रेक के अंदर हम आप लोगों को थोड़ा सा एक्स्ट्रा क्लास प्रोवाइड करेंगे, तो प्लीज आप ना उसके अंदर मार्क कर दीजिएगा अपना समर ब्रेक कब से लेकर कब तक है. ठीक है सर, मैं एक बार स्क्रीन शेयर कर दूंगा, आप एक बार को होस्ट बना दीजिएगा. बस सर 2 ही मिनट लगेंगे मुझे, मैं बच्चों को एक बार बता देता हूँ, बाकी फिर आप कंटिन्यू करिए.
एक सेकंड.
सर आपको ना मुझे को होस्ट भी बनाना होगा सर.
ओके.
एक्चुअली मैं दूसरी आईडी से आज ज्वाइन कर गया हूँ. बच्चों बस 2 ही मिनट लगेंगे, ज्यादा कुछ खास है नहीं, बट ये थोड़ा जरूरी है ताकि हम आप लोगों के लिए कुछ क्लासेस प्लान कर सकें समर्स में.
ये मोहित गुप्ता इज अ सेपरेट पर्सन क्या सर?
यस मेरा मोहित कुमार आपको सर दिख रहा होगा अभी.
ठीक है, हो गया सर, आपको पोस्ट कर.
तो अभी ना मैं बच्चा आप लोगों के सामने स्क्रीन शेयर करके बस आप लोगों को बता देता हूँ कि वो आपको ब्रेक मतलब सॉरी गूगल फॉर्म किस तरीके से फिलअप करना है। बस हमें थोड़ी सी आप लोगों के लिए एक्स्ट्रा क्लासेस मार्क करनी है, तो उसके रिगार्डिंग ये फॉर्म है। देखिए आप लोगों के सामने स्क्रीन शेयर हो गई है, तो ये समर ब्रेक का आपको दिखेगा FI समर ब्रेक टाइम स्लॉट। और अगर कोई उत्तराखंड का बच्चा है, शायद एक उत्तराखंड की बच्ची भी हमारे पास में आती है यूके की बच्ची इस वाले बैच में, शायद अभी ज्वाइन किया होगा, तो ठीक है, तो यहाँ पर यूके लिखा होगा बस। बाकी अगर आप ड्रीम कॉलेज प्रोग्राम के स्टूडेंट हैं तो आपका यहाँ FI लिखा होगा, यानी कि FI मीन्स फीडिंग इंडिया, बस आप ये चीज याद रखें। आप लोगों को ग्रुप में ही फॉर्म मिलेगा, तो वो ग्रुप के हिसाब से जाएगा। नेम ऑफ द स्टूडेंट कि आपने हमारे यहाँ किस नाम से एनरोल कर रखा है। रजिस्टर्ड फोन नंबर, आप लोगों का रजिस्टर्ड फोन नंबर क्या है हमारे पास में। नेक्स्ट आप लोगों का कौन सा स्ट्रीम है, जैसे कि आप सभी लोग पीसीबी के हैं यहाँ पर, तो पीसीबी आप लोगों को चूज कर लेना। यहाँ पर दो चीजें हमें ध्यान रखनी है। योर समर वेकेशन स्टार्ट ऑन, यहाँ पर जब आप क्लिक करोगे तो एक कैलेंडर ओपन हो जाएगा मोबाइल में, जो अभी आपको यहाँ पर नहीं ये रहा। बस आपको ये देखना है कि सर मई 2024 या फिर आपको थोड़ा सा बस बैक जाना है, यानी कि अप्रैल 2024, यानी कि आप लोगों के कब से समर ब्रेक स्टार्ट हो रहे हैं। स्टार्ट आपको बस डेट चूज कर लेनी है कि सर मेरा तो अप्रैल में स्टार्ट हो गया था 25 अप्रैल से, या फिर सर मेरा तो मई में भी स्टार्ट होने वाला है, जो भी मई से आपका स्टार्ट हो रहा है। नेक्स्ट आपका समर ब्रेक एंड कब हो रहा है, तो यहाँ पर आप लोगों को फिर से अपना डेट चूज कर लेना, मई में कब खत्म हो रहा है या फिर आप लोगों का जून में कब खत्म हो रहा है, बस ये आप अपने अकॉर्डिंग देख लेना। समर ब्रेक लास्ट में यहाँ पर आपको लिखा हुआ है कि व्हाट इज योर एक्साइटमेंट फॉर द एक्स्ट्रा प्रैक्टिस समर क्लास, यानी कि अब ये आप सभी का हाई आने वाला है कि आप समर में चाहते हैं ज्यादा से ज्यादा एक्स्ट्रा प्रैक्टिस समर क्लासेस लगा सकें। ठीक है, बाकी आप अपने अकॉर्डिंग मार्क कर देना, बट यहाँ पर हमें लग रहा है कि सभी बच्चों से काफी कॉलिंग की बात हुई है, तो काफी सारे बच्चे डिमांड कर रहे हैं सर एक्स्ट्रा क्लास लगा दो, हम लोग खाली हैं तो ज्वाइन कर पाएंगे। नेक्स्ट ये दोनों क्वेश्चन बहुत इंपॉर्टेंट है, लास्ट वाला यहाँ पे लिखा है योर प्रीफर्ड क्लास पैटर्न ड्यूरिंग समर, यानी कि आप अपने समर में जो अभी क्लासेस चलने वाली है वो कैसे चाहते हैं। 2 क्लासेस ऑफ 1.5 आवर्स पर डे, यानी कि आपको 2 क्लास चाहिए दिन में 1.5 घंटे की, या फिर 2 क्लासेस ऑफ 1 आवर पर डे, यानी कि 2 क्लास चाहिए सर 1-1 घंटे की, यानी कि आपके 2 सब्जेक्ट हो जाएंगे 2 क्लास के अंदर, 1-1 घंटे के 2 सब्जेक्ट। यहाँ पर फिर आपका प्रीफर्ड टाइमिंग स्लॉट क्या है कि आप चाहते हैं कि एक क्लास आपके आफ्टरनून आफ्टरनून में लग जाए और एक आपकी इवनिंग में, या फिर आप चाहते हैं कि दोनों इवनिंग में लग जाए। ठीक है, अगर किसी बच्चे को कोई डाउट है तो अभी आप आस्क कर सकते हैं, बाकी क्लास के बाद में ये लिंक आप लोगों को ग्रुप में मिल जाएगा फिल करने के लिए क्लास के बाद। ठीक है, अगर किसी को कोई डाउट है तो आप लोग पूछ सकते हैं, और अगर डाउट नहीं है तो फिर आप क्लास को कंटिन्यू कर सकते हैं। ठीक है, चलिए सर मैं हटा देता हूँ अभी स्क्रीन को, आप कंटिन्यू कर लीजिएगा। थैंक यू सर।
Thank you. Thank you. All right. So I guess everyone can see the screen. Okay. Yeah. So now you have, so that is something you have to know about the carpel. You'll have ovule. Ovule will turn into the seed in future, and ovary will turn into the fruit. Okay. Now, so you can see over here you have a stigma. That's a landing platform for pollen grain. A very simple thing you can see. Stigma आपका वो area है. So this is my stigma. यहाँ पे मेरा pollen grain आके land करेगा, right? Then you have a style, which is a long stalk-like structure. You can see the definition that been given over here. So style is a long standard part below the stigma from where the pollen tube makes an entry into the ovary. So यहाँ से आपका pollen tube travel करके नीचे तक जाएगा. And then you have ovary. The swollen base part of the pistil. Fertilization occur inside the ovary. So आपका fertilization ovary के अंदर ही होता है. याद रखना, always inside the ovary you will have the ovule, right? Always within the ovary you will have a presence of the ovule, और ovule के अंदर आपका सारे events आपके यहाँ पे होंगे. So now when we were discussing something in terms of the thalamus is the base part. जो base का जो आपका part है यहाँ पे आपका, जिसके ऊपर आपका ovary लगा हुआ है, this is my thalamus, right? So you can see you have a style, you have ovary. Okay, now the thing is that when we discuss male gametophyte, तो वहां पे हम discuss करते हैं, you have something like your anther, right? So you have an anther, you have a bilobed anther, and then you take a cross section of the anther, and when you take a cross section of the anther, you get to see this 4 sac-like structure. You will get to see इन 4 sac-like structure को हम लोग क्या बोलते हैं? What do you call them as a
tetragonal?
Tetragonal is a 4. What is the structure actually? What do you call them? इतने जल्दी कैसे भूल गए? ये जो बेटा स्ट्रक्चर ये जो है ये क्या है?
ठीक है, Thika.
हाँ ठीक है, थीका, ओके ठीक है. सो यू हैव लाइक सिंगल लोब विल हैव टू थीका. ओके, गॉट इट. ये थीका का एक और नाम क्या होता है? व्हाट इज दैट वन थिंग व्हिच वी कॉल इट एज अ
पोलन सैक।
पोलन सैक. ठीक है, पोलन सैक एक आपका नाम होता है. एक और नाम क्या होता है पोलन सैक के अलावा? एक और नाम यहाँ पे क्या होगा?
माइक्रोस्पोरेंजियम।
Exactly, you will have a microsporangium. So it is also called as my microsporangium. Right. So now you have a term like a microsporangium, which we will be using in this particular male gametophyte. Similarly, you have a term like a megasporangium. जैसे आप यहाँ पे microsporangium हम यहाँ पे term use करते हैं, उसी तरीके से you will have one more term, and that term over here is called as my megasporangium. Now, megasporangium is a term which will be used for my ovules. So याद रखना, megasporangium is a term which we'll be using for the ovule. So ovule के लिए जो हम term use करते हैं, that is going to be megasporangium. So that should be clear to all of you that megasporangium is nothing but my ovule, right? And इस ovule के अंदर ही आपका हम बनाएंगे यहाँ पे हमारा megaspore. So in ovule only we'll be making the female egg will be formed within the ovule. So we'll see how that has been formed, but you have to know this thing very clearly that megasporangium is another name for my ovule. So that should be clear to all of you. Okay. Now, so inside the ovary is a cavity known as a locule. Placenta is present inside the locule from which the megasporangium or ovule arises. Now, if you just happen to see, so what we'll get to see now, let's say this is my carpel. So you have a long stalk and then you have a, ovary over here. Okay, you have an ovary in the bottom area. Now what happens inside the ovary? You'll have the ovule. Inside the ovary, you'll have presence of the ovule. Now if you just randomly make a ovule over here and just make a big ovule in this, so this ovule will be attached at the base of the ovary. This ovule will be seen attached at the base of the ovary. So what do you get to see now? What you will get to see that assume that this is my, let's say this is my ovule. So this ovule will have its base attached to the wall of the ovary. So assume that this is my ovule over here, right? So it will be attached to the ovary wall. ये आपका ओवरी के वॉल से, ये ओवरी है मेरी यहाँ पे और ये ओवरी का यहाँ पे वॉल है और उससे ये अटैच्ड है, राइट? सो assume that this is my ovary wall और ये ओवरी वॉल से मेरा यहाँ पे अटैच होगा. This will be attached to the ovary wall over here. So this particular portion from which this particular ovule has been attached, this particular area is called as my placenta. This particular part over here will be called as my placenta. So this ovary wall, जहां से मेरा ovule attach होता है, this part over here is called as your placenta. So that is something to be clear to you that this is something which will be called as your placenta over here, right? So याद रखना, your ovule will be always attached to the placenta, right? And then you will find the kind of arrangement in the ovule will be seen in the, that is going to vary from plant to plant, that how the ovules will be arranged over there. Now let's try to see them in a picture over here. See, so when we discuss the gynoecium, what you get to see that in gynoecium you can have either a single carpel. So you have a flower and a flower will have a single carpel. So that condition is called as my monocarpellary. So if you have a flower and the flower has a single carpel, then you call it as a monocarpellary. That means it has a single carpel or it has a single pistil. It has a single pistil over there. Now, if you have a condition where you have multiple pistil, एक pistil नहीं है, ऐसे 7-8 pistil वहां पे लगे हुए हैं. एक single flower में if you have more than one pistil, if you have like multiple pistil or multiple carpel, then that is called as my multicarpellary. That simply means that you have more number of pistils. So more than one pistil can be seen in the flower. So that can be seen. Sometime you can have a flower which will have like only one pistil. Sometime you can come across a flower which will have like 4, 5, 6 pistil. So that can also happen. So you have a two terms for that. You have a monocarpellary and you have a multicarpellary. So these are the two terms you have to know first of all, right? Then assume that you come across a flower and a flower has 4 pistil. Flower has 4 pistil and those 4 pistil are free from each other. So assume that you have 4 pistil. You have a pistil 1, you have a pistil 2, you have a pistil 3, right? Such you have like 4 pistils are there, and all those 4 pistils are like detached from each other. They are free from each other. So if they are going to be free from each other, if all those particular 4 pistils, assume that these are my 4 pistils, and these pistils are free. If these pistils are free, then such condition over here will be called as my apocarpus. Such condition can be called as my apocarpus. When you have a pistil and they are going to be free from each other, then such condition can be called as my apocarpus condition, right? On the other hand, if you have all those pistils, right, you have 4-5 pistils and they are all attached to each other, सबके सब एक दूसरे से चिपके हुए, if all those pistils are been attached to each other, then that condition over there is called as my syncarpus. Then that condition over there will be called as my syncarpus. So syncarpus is a condition in which you will find the pistils are been attached to each other, and apocarpus is a condition in which you will find the pistils are going to be free from each other. So yaad rakhna, syncarpus mein you will find the pistils are attached. So pistils are attached. So they will be all attached to each other, and if at all you happen to find that thing happening, then yaad rakhna, that is my syncarpus condition. So these are the 2 terms you have to know, syncarpus and apocarpus. These are the terms should be clear to all of you. ये term clear है आप सबको यहाँ पे? Is this term clear to all of you? All right, ठीक है. All right, got it. Okay, then. So यहाँ पे लिखा हुआ है apocarpus means carpels are going to be free, and carpels if they are fused or attached, that is my syncarpus. So you can see over here you have a syncarpus example. Papaver is one of the plant where you can find all the pistils are going to be attached to each other. That's my syncarpus condition. And Micaelia is going to be one of the plant in which you will get to see that your apocarpus condition, where the pistils are going to be free from each other. They are not attached, they are going to be free from each other. So they are not attached over there. So that is my apocarpus condition. So that is something you can get to see in the plant. These are the same example you will also find in the NCERT as well. Okay. Now let's talk about my ovules. Right now, when we discuss ovules in detail, now because we don't have to get into a very detail of the ovules, but right now you can see that ovules are been seen of my different types. So ovules which you get to see in the plant are going to be of your different types. Okay. Now just let me draw one over here. When you observe an ovule, what do you generally get to see? Right. So this is an ovule, and as we have discussed, this will have a long stalk, and this stalk will be attached to the ovary wall. This stalk will be attached to the ovary wall, and the ovary wall will be having a small bulging structure on the ovary wall, and that bulging structure ko hum kya bolenge? What was the name of this bulging structure over there? Itne jaldi
Placenta.
placenta. Right. That is my placenta over here. Yeah, that's your placenta. Right. So I have a placenta over here that's present on the ovary wall. Right. You have this long stalk-like structure. That stalk-like structure is called as my funicle. That stalk-like structure over here is called as your funicle. Right. And you have this particular ovule. And in this ovule, what you will get to see, you have this narrow opening over here. This narrow opening in this location will be called as my micropyle. So you have this narrow opening here, and this narrow opening here is called as my micropyle. Right. Now, what do you get to see? Now, when you observe an ovule, now you will have some outer coverings. Ovule will always going to have a presence of some outer coverings. So we will always have a presence of some 2 or 3, generally 2, you will have a presence of the outer coverings. So these outer coverings of the ovule are called as my integuments. These outer covering of the ovules are called as my integuments. Right? So 2 to 3 layers of integuments you get to see in the ovary. Right? Then, so these are the outer covering of the ovule. Now, what you get to see inside the ovule? Now, ovule inside you will have some group of cells. So inside the ovule, you will have a presence of some group of cells, and whatever the cells, now I'm not going to draw all the cells, but whatever the cells you find inside the ovule, now those cells have a name, and that name is going to be called as my nucellus. So nucellus are nothing but my group of cells which you find inside the ovule. So that is what you will get to see in the ovule, right? You have a funicle, you have a placenta, right? And you have the outer covering of the ovule is called as my integuments, right? So you can have a 2 layers. Sometimes you can also find 3 layers, but yeah, 2 layers are very commonly seen in the outer layer covering of the integuments. Then you have one small opening. That small opening is called as my macropyle, right? And you can also know one more thing over here. Opposite to macropyle, you have a region, and that region here is called as my chalaza. So opposite region of a macropyle is called as my chalaza. So now let me just complete the ovule. ऊपर से ऐसा खुला नहीं होता, ये बंद होता है यहाँ से. So yeah, that's one thing. So let's close the ovule over here. Right. So the opposite end over here will be called as my chalaza end. So if they ask you what has been seen opposite to macropyle end or macropyle, their answer will be the opposite end is will be my chalaza end. So chalaza has to be the opposite end of the macropyle. So that is also one thing you have to know over here, right? So you have a chalaza, you have a macropyle. Okay. Now, this is a type of ovule you very commonly get to see in the angiosperms. So in the angiosperms, you get to see this kind of ovule will be seen, and this ovule is called as my anatropous type of my ovule. So this ovule you can see in front of you. इसको हम बोलते हैं यहाँ पे as a anatropous. You call it as a anatropous. So anatropous type की ovule आपको मिलेगी generally देखने के लिए आपके angiosperms के अंदर काफी commonly. Where what you get to see, it is inverted. मतलब it is been inverted. मतलब ये like see funicle के साथ ये parallel है और ये inverted है, उल्टी है ये. Inverted हम क्यों कह रहे हैं? Why we are saying that it is inverted? Because the macropyle is facing towards the downward direction. So macropyle is facing towards the downward direction, and it has been very much parallel to your funicle. So it is going to be anatropous and going to be inverted. So anatropous plus inverted. These are the two terms you can know. So anatropous ovules are always going to be inverted, right? So this is what you will get to see. Now, if I show you a picture, if I show you this picture, you have a different types of ovule which you can to see in the plants. You can very clearly see the anatropous over here, right? You can very clearly see the anatropous over here. Okay, and all the time, or like all these different kind of ovules which you can see in front of you, you will never get a question based on any other ovule. You just have to know everything in terms of the anatropous. So anatropous is an inverted ovule where you get to see that my macropyle is facing towards the downward direction, right? And it has like it has been very much parallel to your particular funicle, right? On the other hand, you have an orthotropous. अगर तुम देखते हो यहाँ पे, so it is facing in the upward direction. So यहाँ पे आपका downward direction में आपका macropyle open हो रहा है. In the orthotropous, you are finding the macropyle is facing towards the upward direction. And orthotropous is a kind of ovule you get to see in the gymnosperms. It is very going to be commonly get to see in the gymnosperms, right? Then if we see the hemianatropous, so here what is happening? The ovule is making a right angle to the funicle. The ovule is actually making a right angle with the funicle, and that is my hemianatropous. अब ये सब आपको ignore करना है. You just have to pay attention to the anatropous kind of ovule. This is the kind of ovule you will get to see in the angiosperm. So that should be clear to you. Baki you can apparently ignore it. There is no, you don't get to see questions coming from them, right? Now, so let's try to understand the structure of your anatropous ovule. Now, as I've told you, you can very clearly see in this picture that when you discuss ovule, you have the outer covering, that's my integument. You have a opposite end, that's my chalaza. Opposite chalaza, you have a macropyle end, right? You have a funicle, you have a placenta. Now, there's one thing you have to understand. The point of attachment where a funicle is getting attached to the ovule, the point of attachment where the funicle is getting attached to the ovule, जहाँ पे मेरा funicle ovule के साथ attach हो रहा है, this point of attachment जहाँ पे मेरा funicle मेरा ovule के साथ attach हो रहा है, this point of attachment is called as my hilum. This point of attachment is called as my hilum. तो ये याद रखना, you will get question based on this. So the point of attachment between the funicle and the ovule is called as my hilum. If they ask you what is that small stalk which connects my ovule with the placenta, then your answer is going to be my funicle. The small stalk which connects my ovule with the placenta, that is called as my funicle. If they ask you what is my nucellus, then you have to tell them all the cells which are found inside the ovule, that is called as my nucellus. And abhi, right now, we haven't made anything inside the ovule. We are just saying that you have some cells inside the ovule. You call them as the nucellus. We are not making something like a megaspore right now. We are not making something over here right now. right? But you have to know the cells inside the ovule is going to be a nucellus. Okay, and please remember, all the cells over here are going to be diploid. Your integuments will be diploid. Your nucellus will be my diploid. So nucellus cells are also going to be diploid over here, right? All the cells over here are going to be diploid. So this is something should be clear to you. Hilum याद रखना, फनिकल याद रखना. फनिकल कनेक्ट कर रहा है प्लेसेंटा को ओव्यूल के साथ. Hilum is a junction between the ovule and the funicle. So I hope इसको लेके कोई confusion नहीं है. Confusion हो तो please पूछ लेना. You should be very much clear with this, right? Now, okay. So this is what you will get to see. So again, this is a NCERT picture over here, right? So you can see this will be my macropyle end. This is going to be your micropyle end. Let's do one thing. You tell me the labeling now. You help me out with the labeling over here. Now, if I ask you that this is my particular ovule and it has like this two outer coverings, you get to see over here. So these two outer coverings have been called as what?
Integuments.
Right, they're called as my integuments. Right, bilkul sahi. They are my integuments. Okay, true. Then tell me, what do you think if that is my macropyle? So this end over here is called as what?
Chalaza end.
Chalaza end, right? This end, yes, it's called as my chalaza end. Okay. Then this long stalk, if I say this is my long stalk over here, then this long stalk is going to be what?
Funicle.
Right. This long stalk over here will be my funicle. Very true. Okay. And if I see over here, this funicle is getting attached to the ovule ultimately. So let's say this attachment, this point of attachment over here, where my funicle is getting attached to the ovule, that point of attachment will be called as what?
Hilum.
Right. Very good. So all of you are true. Okay. So have a look at this picture over here. You can see the chalaza end, you can see the macropyle end, right? Ignore the, in the center you have embryo sac, usko ignore karo. Usko hum banayenge. Abhi wo hai nahi. Right? You have integuments, you have a two outer covering over here, you have integuments, and you have a nucellus. Nucellus are nothing but all the group of cells you will find inside the ovule. So please get yourself very clear with the picture. And it is my anatropous inverted ovule. Right? You can see the hilum, you can see the point of attachment as my hilum, you can see the funicle, you can see everything which have been mentioned over here. Right? Now, let's forget the embryo sac right now. Let's forget the raphe over here. We'll see them later. Abhi hame embryo sac banana hai. Now, so again, I'm just, the pictures are coming multiple times in front of you so that you remember them. What is going to be seen where? That should be clear to all of you. Okay. Embryo sac will be seen in the center. How that is getting formed is now something we'll try to see. Okay. So first of all, let's read all those things. Funicle is what? Ovule in the ovary are attached to the placenta with the help of the funicle. Something I've already discussed. Hilum, junction between the ovule and the funicle. Junction between the ovule and the funicle is called as my hilum. Integuments, inner and outer integuments surround the nucellus and provide the protection to the developing embryo. So these are my two layers which are known to provide the protection to the developing embryo. That's my integument. Nucellus, layers surrounding the embryo, function provide nourishment to the developing embryo. So nucellus is nothing but all the group of cells you will find in the center, and they're going to play the role in providing the nourishment to the embryo. Whatever the embryo is developing inside, usko nourish वहाँ पे करेंगे. हाँ जी बेटा बताइए. मोहित बताइए. Someone has asked the question. Yeah, Tanishq, tell me.
So sir, nucellus is analogous to placenta.
Nucellus analogous to placenta. Yeah, the placenta you get to see in the human beings. Right. Yeah, you can say that. Now, so now you have over here the macropyle. Integuments enclose the ovule except a small opening where the pollen tube makes an entry, which is called the macropyle. So the macropyle which you were seeing over here, this is a location through which you will be finding the pollen tube is going to enter inside. This is a site from which you will find the pollen tube is going to enter inside. So that is something you have to know. That's my macropyle. Okay. Now we'll jump onto the story of my megasporogenesis. Now let's talk about my megasporogenesis over here. So that how that will be happening. So the formation of megaspore from the megaspore mother cell via meiosis is called as my megasporogenesis. Only one of the megaspore remains functional and forms the embryo. Rest of megaspore are going to degenerate. So let's forget that and let's try to understand this thing. So let me try to show you a few things over here. Okay. Now what you find happening over here now? assume that this is my ovule. This is your ovule over here, right? Okay. Now inside, you have your nucleus, right? You have your nucleus. You have all the nucleus inside the ovule. Now what do you get to see? One of the cell in the nucleus will become enlarged. One of the cell in the nucleus, and the cell is generally seen nearby the macropyle end. So one of the cell in the nucleus will become enlarged. So समझो यहाँ पे cell है, that will become enlarged. And you know that all the cell in the ovule are going to be my diploid cell, right? Now one cell in the nucleus which will get enlarged can be called as my megaspore mother cell. This can be called as your megaspore mother cell. This will be my megaspore mother cell. Okay, so this will be your MMC. Now what happens? Now what is the story of this particular MMC over here? Now this megaspore mother cell which you have inside the ovule, this megaspore mother cell will undergo meiosis. And once it undergoes meiosis, you will find the 4 megaspore are being formed. So you know that the outcome of meiosis is always going to be your 4 megaspore. So जैसे मेरा diploid cell यहाँ पे meiosis करेगा, you will have formation of 4 megaspores will be seen. So 4 megaspores will be formed after the meiosis. And all these 4 megaspores which have been formed over there are going to be haploid. You know that now after meiosis, whatever you get over there is going to be a haploid cell. So these 4 megaspores have been formed, and these 4 megaspores are going to be haploid in nature. Right? Now what happens? When you see this 4 megaspore, out of this 4 megaspore, 3 of them will undergo degeneration. Out of this 4 megaspore, you will get to see 3 of them will undergo degenerate. So they are going to degenerate. So these 3 megaspore are going to degenerate, and only one survives. Which one survives? Someone who is going to be closer to the macropyle end again is going to be the one which is going to survive, right? So one of the megaspore is going to survive. So this is my one megaspore which has fortunately survived over here. So that's the first thing you have to know. And the next thing you have to know is this megaspore is the one through which you will form the complete embryo sac. This megaspore is something which I'll be using to form the complete embryo sac. कैसे बनता है वो हम देखते हैं. But first of all, you have to know, using this megaspore, I'll be finding the complete embryo sac now will be formed. So using this, you are going to form the complete embryo sac. How that will be happening? Now let's try to understand that how this will be happening. But this is something you have to know. So again, yaad rakhna, inside the ovule, you have a nucleus cell. One of the cells of the nucleus will enlarge in size and will behave like a megaspore mother cell. That megaspore mother cell, which has been drawn over here, will undergo the meiosis, will give rise to my 4 megaspore. Out of this 4 megaspore, 3 are going to degenerate, 1 survives, and that 1 megaspore which is going to survive will give rise to a complete embryo sac. Okay, that will give rise to embryo sac, and the embryo sac itself is been called as my female gametophyte. Embryo sac का एक और नाम यहाँ पे आपको हो जाता है female gametophyte. So embryo sac has been also called as my female gametophyte. ठीक है, कोई भी अगर दिक्कत आ रही हो देन इंटरप्ट करके पूछ लेना. If there is something you don't understand, if there is something you want me to repeat again, then please let me know that. So this embryo sac which has been there, which is now also can be called as my female gametophyte. But अभी हम देखेंगे कि बनता कैसा है. How one megaspore can lead to formation of such embryo sac. So now you can see we have taken one of the cell inside the ovule as a MMC. And this MMC now will undergo the meiosis and will give rise to a tetrad. You can see a megaspore tetrad can be seen. So you can see over here these 4 which have been formed over here. These are nothing but my megaspore tetrad. Megaspore tetrad. Do remember these megaspore tetrad, once they have been formed, like you will find 3 of them will degenerate and 1 of them is going to survive. So that should be clear to all of you. Okay. So that is what you will get by the end of the meiosis. So you can say this is my, let's say this is my meiosis 1, and then you have a meiosis 2. And after that, you have this. After the end of the meiosis, you get to see this is something which has been formed. Okay. Now let's see. Now let's understand how this megaspore, a single megaspore, is going to make everything inside the ovule. Right. Now Assume that you have an ovule over here. You have an ovule again. We made a slightly bigger micropyle. हमने यहाँ पे बना दिया है, काफी बड़ा बना दिया. थोड़ा कम करते हैं. Yeah. Okay. Now assume that you have your, now all of them have degenerate. So now your 3 megaspore have degenerate over there, and now you have your single. Let's say you have your single megaspore over here. Now what happens when you have a single megaspore? In this single megaspore, it will undergo 3 times mitosis. It will undergo 3 times mitosis. So 3 times mitosis will be seen. So 3 times mitosis is observed. Now, but this mitosis which will be observed over here will be slightly different. Now, what actually happens when you say that 3 times mitosis will take place with this particular single megaspore? It will be my nuclear mitosis. So, what do you get to see? The nuclear mitosis will be seen. That means cytokinesis will not take place, but the number of nucleus are going to increase. So, nuclear mitosis will be seen happening over there. So, you will get to see nuclear mitosis will be seen taking place inside the megaspore. So that means what? You have a single megaspore, and suddenly what will happen? There is no cytokinesis taking place. Only nucleus is getting divided. So nucleus का number यहाँ बढ़ेगा, और याद रखना there will be no cytokinesis. There will be no cytokinesis. Now, if you have a condition where no cytokinesis is taking place, आपका cell cytoplasm division आपका हो नहीं रहा, सिर्फ आपका न्यूक्लियर डिवीजन आपका यहाँ पे हो रहा है. कितने बार? 3 टाइम्स माइटोसिस होगा, और वो भी किसका होगा? न्यूक्लियस का होगा. 3 times mitosis is taking place, and that too for a nucleus. So now what will happen? If you observe inside, what you will get to see, that assume that this is my now my large megaspore, and now you will get to see you have 8 number of cells. If you 8 number of nucleus, if you undergo 3 times mitosis for the nucleus, then what will happen? The number of nucleus you will have is going to be 8. So now suddenly you have a large number of nucleus can be seen inside that particular cell. So such how you will get to see 8 nucleated cell can be seen over here. So right now you can see over here 8 nucleus will be seen. So you will get to see 8 nucleus are observed. Now once the 8 nucleus have been formed, once these 8 nucleus have been formed, what will happen? These nucleus will get themselves arranged inside that particular area. So now they will get themselves arranged in a certain area. So what happens over here? What you will get to see? That you will find these 8 nucleus will find their way and pick up certain locations in that particular ovule. So what do you get to see? That assume that this is the cell, right? So now what you do is you have a 8 nucleus over there. You will find 3 of the nucleus will get in this particular area in near the chalazal end. 2 nucleus will come in the center, and remaining 3 nucleus will be seen nearby the micropyle end. Remaining 3 nucleus will be seen nearby the micropyle end. So we'll have one. I'll just draw one of a different color. एक हम अलग कलर का बनाएंगे यहाँ पे थोड़ा सा, right? And remaining 2 of the same color. So these are the nucleus will get over, and they will get themselves arranged in a certain location. So now you can see if they ask you कि chalazal end पे कितने nucleus मुझे यहाँ पे आ गए, 3 आ गए. So chalazal end पे आपको you can see some 3 nucleus getting arranged over there. That is my chalaza end. Macropyle will also have the 3 nucleus, and in the center you also have a 2 nucleus over here. So this is how I'll be finding my 8 nucleus will get themselves arranged inside the particular ovule. Now you have to know, once these nucleus are getting themselves arranged at certain locations, to find the wall formation can be seen. Once the nucleus have got themselves arranged in a certain locations, now you get to see the wall formation will be seen, or I can say now cell wall formation will be observed. cell wall formation is seen. Now, if you see the cell wall formation happening over here, so this nucleus will get a cell wall, so that becomes a separate cell. This will get a cell wall, it becomes a separate cell. This also gets a cell wall, so there is wall formation, that will become a cell. Right? Then you have over here, you have one more cell getting formed over here. Just wait a minute. You have one more wall formation to this, and I have one more wall formation to this. So now you have how many cells now you can see over here? 1, 2, 3 at the top, and 3, 4, 5 at the bottom. Okay. So now you can see 6 cells can be seen over here. Will the 2 nuclei in the center will get any kind of wall formation? The answer is no. जो बीच में जो मेरे यहाँ पे 2 न्यूक्लिया है उनके अंदर आपको वॉल फॉर्मेशन आपको नहीं मिलेगा. They won't have their own separate wall over here, right? So what you have to understand here is, now what you get to see that at the chelazal end you have like 3 cells. At the macropyle end also you have 3 cells. At the macropyle end also you have 3 cells, and in the center you will have a presence of a 2 nuclei. You will have a presence of a 2 nuclei, and please remember all of them are my haploid cell. याद रखना सबके सब haploid है. Haploid क्यों है? Because they are coming from the megaspore. They are all coming from the megaspore, and megaspore was always going to be my haploid. You know that now. Megaspore was always going to be haploid because it is coming from it is the outcome of a meiosis. So that is also going to be haploid megaspore, right? तो इसमें से ही हमने सब कुछ बनाया है. तो जो भी मेरा यहाँ पे बनेगा, that is going to be my haploid only. So याद रखना, a megaspore will undergo 3-time mitosis, and that will be my nuclear mitosis. That means cytokinesis will not be observed. And after nuclear mitosis, you will have the 8 nucleus will be formed, and this 8 nucleated structure can be seen over there getting formed, and these 8 nuclei will get themselves arranged at a certain locations. Once they get themselves arranged in a certain locations, there will be a cell wall formation which will be observed. Once the cell wall formation can be seen happening over there, then you can see you have a 3 cell at the top, 3 cell at the bottom, right? And you have one more cell, this large structure which I've drawn. This is called as my central shell. This cell, which is a very large one, this is called as my central cell. This is my central cell. So 3 cells at the top, 3 cells at the bottom, and one large cell, that is my central cell. So कितने हो गए मेरे यहाँ पे total अभी यहाँ पे? How many cells now I have?
7 cells.
7 cells I can see. How many nuclei you can observe in these 7 cells?
8.
Right. You can see the 8 nuclei. Right. So that is why I can say it is my 7-celled, 8 nucleated embryo sac. 7-celled, 8 nucleated embryo sac. So this is what you will get to see in the embryo sac. You have a 7-cell structures and 8 nucleated embryo sac will be observed. This is what you will get to see in the embryo sac. And whatever we have formed over here is nothing but my embryo sac. तो ये जो हमने यहाँ पे बनाया ना अभी, this is called as my embryo sac. ये मेरे यहाँ पे embryo sac. तो अगर आपसे कोई पूछता है randomly, if your friend asks you how many cells you find in the embryo sac, the answer should be 7. If they ask you how many nuclei will form in the embryo sac, the answer will be my 8. Right? So 8 nuclei and 7 cell structure can be seen in the embryo sac. Okay. Now there's one thing you have to know. We have to remember the name of the cells. Now we have to know what are these cells are called as. पहले ये बताओ यहाँ तक समझ आ रहा है नहीं आ रहा? Is this thing clear to all of you? Tanishq, Misbah, Gayatri, Aisha. I hope it's clear to all of you. Okay. And all the other people who are here. Shikha, it's clear to you as well.
Yes,
I hope मैं
sir.
किसी को miss नहीं कर रहा. ठीक है. And Mohit Gupta, if you are there in the class. Okay. Right. So that is something you will find happening over here. This is what you get to see. Now the 3 cells at the chelazal end, the 3 cells you are getting to see in the chelazal end, these 3 cells have a name and they are called as my antipodal cell. They are called as my antipodal cells. Okay. That is something you have to know. These cells are called as my antipodal cells. Now at the bottom we will get to see, at the bottom we will observe, we have these 2 cells which I have drawn separately other than the orange one. These 2 cells over here are called as my synergate cells. These 2 cells over here, these 2 cells have a name and these cells are called as my synergate cells. These are my synergate cells. Okay, this orange cell is actually going to be your egg cell. This orange cell over here is called as my egg cell. This is the egg. ये है वो egg जिससे आगे sperm bind करेगा. ठीक है, this is my egg cell. So इसलिए उसको हमने color अलग दिया. That's my egg cell, and you have my two synergate cells can be seen over there. Egg cell and synergate cell are been collectively called as my. They are collectively given a name, and that name is called as my egg apparatus. That has been called as my egg apparatus. So याद रखना, इनको हम बोलेंगे यहाँ पे as a egg apparatus. So if someone asks you कि egg apparatus क्या होता है, कोई अगर आपसे पूछे कि क्या होता है egg apparatus, तो आपने बोला है कुछ नहीं होता. It will have the egg cell and a synergate cells. That is what I'll get to see in the egg apparatus. Okay. Now also remember one more thing. If I know this much amount of thing, also know one more thing, that nearby the synergate cells, you will have a finger-like projection. Nearby the synergate cells, you will have some finger-like projection can be seen. This finger-like projection you will find nearby the synergate cells are called as my filiform apparatus. So कुछ finger-like projection आपको यहाँ पे मिलेगा, and that finger-like projection is called as my filiform apparatus. What it does वो हम बाद में देखते हैं, but याद रखना this is called as my filiform apparatus. Seen nearby your synergate cells. हाँ जी गायत्री बताइए, बोलिए गायत्री बोलिए.
Sir, due to network problem, video was off, sir. Sir, can you repeat, sir?
Which part has to be repeated?
Sir, after the 8 nucleus and 7 cells in embryo sac, after that.
Okay, got it. Right. So once you have this 7-celled and 8-nucleate structure has been formed, which is called my embryo sac. This is nothing but my embryo sac which has been formed. Now we have to name them. So the 3 cells at the chalazal end are called as my antipodal cell. The 3 cells which you find in the chalazal end are called as my antipodal cell. Then you have a 3 cell at the bottom. The 3 cells at the bottom are going to be, out of those 3 cells, 2 cells are called as my synergate cells. These are called as my synergate cells. Right? You will have synergate cells at the bottom nearby the macropyle end, and along with that you will also have your egg cell. Along with that, the orange one is going to be your egg cell over here. So egg cell and a synergate cell are the one which are collectively called as my egg apparatus. You have a name for them, and they are being collectively called as your egg apparatus. So egg apparatus will be having 2 things. You will have the egg, which is the female egg over there, and you have a synergate cells, and these 2 collectively are called as my egg apparatus. Along with that, you have to know there will be some finger-like projection can be seen over there above the synergate cells. That is been called as my filiform apparatus. That is called as my filiform apparatus. Now the only thing remaining right now is going to be, you have these 2 nuclei in the center. These 2 nuclei in the center are going to be called as my polar nuclei. So you have these 2 nuclei in the center, and these 2 nuclei in the center are been called as my polar nuclei. They are called as my polar nuclei. So these are the labeling which you should all remember, because you will find this question coming on based on all these labelings. Now let's try to see few things over here. So now so that is something you cannot see right now, but this is what the 8-cell structure has been formed now. Okay. So this is how the mature embryo sac is going to look like. This is the mature embryo sac is going to look like. So now you can see the mature embryo sac, and here this where the picture is getting overlapped. You have a 2 cells, then it becomes the number of embryos, and then you have a 2 nuclei after single mitosis, and you have 3 times mitosis, then you'll have a 8 nuclei, and then they will get themselves arranged in the particular cell. They will get them arranged in the embryo sac. Okay, now have a look. So now this is my mature embryo sac, or what you also call as a female gametophyte has been formed, right? Now you can help me out in labeling. अब आप labeling में बताओ मुझे यहाँ पे. So Tanish, you tell me these 3 cells at the top are called as my
Antipodal.
Right. These are my antipodal cell. Misbah, you tell me. You have these 2 nuclei in the center. This nuclei are called as my what?
Polar.
Right. They are called as my polar nuclei. Okay. Right. Then you have over here slightly different arrangement. You have these 2 cells and they have a finger-like projection. So these two cells which you can see over here, Aisha, tell us which are these two cells are called as my. आयशा नहीं बताएगी तो फिर कौन बताएगा? शिखा आप बताओ, ये दो सेल का नाम क्या होगा, क्या होगा यहाँ पे? Can't hear you. Synergate cells are the one which are the two cells which you'll get to see over here, and then you have the one is going to be my egg cell over here. This will be my egg, and egg and synergates are the one which are collectively called as what? What you collectively call them as a?
Egg apparatus.
Right, you're going to call them as a egg apparatus. Right, and you can see over here the finger-like projections can be seen. So this finger-like projection which you can see over here, now please understand this thing. You can see this finger-like projection which are called as what?
Filiform apparatus.
Call them as a filiform apparatus. Now please remember one thing very carefully over here, that when we discuss about the filiform apparatus, they have one role to be played. They play one very significant role, and that role is going to be guiding the pollen tube. So ये बात याद रखना, they have a one role to be played, and that one role is going to be your guiding the pollen tube, guiding pollen tube. So whatever the pollen tube is going to come towards the macropyle end is going to come because of them. They are going to release some chemicals. They are going to release some chemical attractant, and they actually help in the guiding the pollen tube. So actually help pollen tube to reach up to the ovule. So guiding the pollen tube. That is what they are being known for. So pollen tube को जो खींच के ला रहा है वो बेसिकली आपके ये फिलिफॉर्म apparatus और synergate cell मिलके वो कर रहे होते हैं. So ये बात आपको यहाँ पे याद रखनी है, because you will find question coming based on them, right? So that is something you can see over here. The picture you can see, you have a synergate cells over here. You can see the filiform apparatus, and you know that collectively what they are being called as my egg apparatus. Also you know that, sorry, synergate cell and the egg are collectively going to be called as my egg apparatus. This is something you know. You have a central cell. Central cell will have two polar nuclei, and you have in the opposite side you have an antibodal cell, right? Yeah, Ms. Bhatt, tell us. बताइए.
Sir, can you show previous slide?
बोलो.
Okay, sir.
Right, so you have a central cell. So these labelings has to be very, very, very clear to you. Chilaza end पे क्या मिल रहा है, macro end पे क्या आ रहा है. You should know. Sometime they will ask you question based on labeling. They'll simply give you the labelings over there, and they will ask you किसको label करो. That can happen to you. So please know all those things. And all the nuclei over here are going to be haploid. Please remember, all the cells and the nuclei in the center are going to be haploid. याद रखना, antibodal cell अगर आपसे पूछे ना कि इसका क्या haploid, तो they are all haploid over here. And why they are haploid? Because they have been formed from the megaspore via mitosis, right? So they are all going to be haploid in nature. Please ये बात हमेशा याद रखना. Okay, so that is something you have seen. So this is where you have actually understood that is my megasporogenesis. So this is what you find happening in my megasporogenesis. Now we can only have a confirmation कि तुम्हें समझ आया by solving some questions. So this is my 7-celled 8-nucleated structure. Egg plus 2 synergate cells are called as my egg apparatus, right? So we can have a look at this later. We have already done this. Let me show you some questions. So ये question नहीं, पहले हम इसके question देखेंगे यहाँ से. Okay, take your time. Take your 30 seconds and then raise your hand to answer this. You have to label this. You have to label this picture. So raise your hand to answer this. A, B, C, D. देखो यहाँ पे. And then raise your hand. All right. Okay. Couple of people have raised their hand. तनिष बताइए क्या लग रहा है इसका आंसर?
सर A part है.
A part. Okay. मिस आपको क्या लग रहा है?
Sir, A.
ये लग रहा है. All right. ठीक है. And क्वेश्चन क्या और कौन था यहाँ पे? शिखा क्या लग रहा है?
Option A.
Yes, option A becomes the correct answer यहाँ पे. So here is going to be the hilum over here, and that hilum is something which is the junction between the funicle and your ovule, right? Then you have a funicle that's going to be a B, which can be seen over here, right? C is going to be the nucellus, okay? And then you have the D1, which is going to be the macropyle. So yes, that's going to be a correct one. The megasporangium of an angiosperm on maturation give rise to. So जो megasporangium होता है वो maturation के बाद आपको क्या बना के देगा? Again ये question कर सकते हो, बड़ा ही आसान question है. मैंने starting में कुछ आपको बताया था. So please raise your hand to answer this. गायत्री इज देयर इन द क्लास और नॉट? गायत्री, मोहित गुप्ता, आप लोग भी ट्राई करो बेटा, सिंपल को आसान है. द मेगास्पोर, अरे यू हैव टू रेज योर हैंड फर्स्ट ऑफ ऑल. हाँ, सो टेल अस मिसबा, क्या होगा इसका आंसर?
Sir, option A, seed.
ऑप्शन ए. मिसबा ऑप्शन ए. ओके, तनिष क्या लग रहा है आपको?
No option.
तनिष.
सर एम्ब्रियो.
ओके, शिखा.
Seed.
सी. अब मेरी बात सब लोग ध्यान से सुनेंगे, सारे लोग मेरी बात यहाँ पे ध्यान से सुनेंगे. सुनो मेरी बात. नाउ टेल यू समथिंग. आई हैव टोल्ड यू वन थिंग. मेगास्पोरेंजियम इज इक्विवेलेंट टू व्हाट? मेगास्पोरेंजियम इज इक्विवेलेंट टू व्हाट?
माइक्रोस्पोरेंजियम.
नहीं. प्लांट के अंदर मेगास्पोरेंजिया जो आपका होता है, एक फीमेल प्लांट के अंदर आपका जो मेगास्पोरेंजिया, वो इट इज सिमिलर टू व्हाट? याद करो, मैंने बोला था मेगास्पोरेंजिया इज सिमिलर टू योर ओव्यूल. बोला था नहीं बोला था?
हाँ यस सर.
बोला था, ऐसे कैसे भूल सकते हो आप लोग? गायत्री, मिसबा, याद आ रहा है ये सब? शिखा एंड आयशा, मेगास्पोरेंजियम इज सिमिलर टू योर ओव्यूल एक्चुअली, राइट? नाउ इफ यू रिमेंबर व्हाट ओव्यूल टर्न्स इनटू, ओव्यूल एंड में बनता क्या है जब आपका मैच्योर हो जाता है ओव्यूल? तो किस में बनता है?
सर सीड में.
सीड में बनता है. Ovule will turn into a seed, बाबा. Your ovary will turn into a fruit and ovule will turn into a seed. लड़कियों को सबको याद आ रहा है ये वाली बात?
Yes,
Yes, sir.
So ovule turns into a seed, and on the basis of that, now you can see the megasporangium, which is actually nothing but an ovule, right, of angiosperm on maturation give rise to. Now tell me what will be the answer over here?
सीड.
Option B. ठीक है, that will be my seed over here. तो सोच के करो, ध्यान से करो, ठीक है? अभी और हम इसे कॉम्प्लेक्स क्वेश्चन आएंगे. एम्ब्रियो सैक डेवलप्स फ्रॉम मेगास्पोर मदर सेल थ्रू. ध्यान से देखो क्वेश्चन को. एम्ब्रियो सैक आपका बनता है, डेवलप्स फ्रॉम द मेगास्पोर मदर सेल थ्रू. कैसे बनता है? सो रीड द ऑल द ऑप्शंस थॉरली एंड देन रेज योर हैंड. गायत्री का क्या नेटवर्क इशू आ रहा है क्या यहाँ पे? मोहित, आपको मेरी आवाज आ रही है? मोहित गुप्ता, क्या आपको मेरी आवाज आ रही है? मे बी नॉट. आयशा, आप भी ट्राई कर सकते हो. गायत्री, डू यू हैव सम इशू विद ऑन योर साइड विद द नेटवर्क और प्लांट गैमेटोफाइट? समझ नहीं आ रहा क्या हो रहा है.
Sir, yes, sir. Due to network problem, the screen was not able to see this.
ओके ओके ओके, गॉट इट. ऑलराइट मिसबा, टेल अस क्या होगा इसका आंसर? मिसबा.
option B.
ऑप्शन डी.
B.
बैटमैन वाला बी. ओके
Yes, sir.
ओके, व्हाट डू यू थिंक तनिष्क?
यस सर बी पार्ट 1 meiosis and 3 mitosis.
एंड शिखा, क्या लग रहा है आपको?
Sir, option B.
Right, option B is going to correct answer. बिल्कुल सही. So you'll have a single meiosis will take place and you'll have a 3 mitosis, and that will be a nuclear mitosis. You won't find any kind of cytokinesis taking place over there. So yes, that is what you will be finding happening over there, and that's how the embryo sac will be formed. So right, very good. Filiform apparatus found during the development in the angiosperm is a thickening on the, this filiform apparatus which you get to see in the angiosperm, ये किस पे लगा हुआ होता है? किसके साथ ही जुड़ा हुआ होता है या लगा हुआ किसके ऊपर ही होता है? Again you can do this. बताइए शिखा, क्या होगा इसका आंसर?
Sir, option D.
एंड तनिष्क, क्या लग रहा है आपको यहाँ पे?
सर synergids option D.
Right. So that is going to be a correct answer. Synergids are going to be the correct answer over here. Yes. Then again, now yeah, I think you all can label this. So I won't be spending time on this question, right? You can do this. This is a question from NEET exam 2000. Okay, now there's one thing I have to tell you. Yeah, there's one thing I have to tell you over here. Let me tell you over here itself. See, now you know from a single megaspore, you will find the complete embryo sac has been formed. The complete embryo sac is getting formed from a single megaspore, which you know you have a 7-celled 8-nucleated structure. Now there's something you have to understand. When a single megaspore is involved to form a complete mega embryo sac, such kind of development is called as my monosporic. Such kind of development is called as my monosporic. So monosporic का मतलब ये होता है कि आपका single megaspore आपका यहाँ पे involve है. You have an involvement of a single megaspore, and that single megaspore is something which will give rise to a complete embryo sac. and that is called as my monosporic. So this condition over here is called as my monosporic, right? So over here, if you see now question, now you can completely answer, very simply answer this, that 8 embryo sac is your only monosporic,
Monospore.
right? So that's something now you can do. चलो अभी वाला देखो, ध्यान से देखना. मेगास्पोर आर प्रोड्यूस्ड फ्रॉम द मेगास्पोर मदर सेल आफ्टर. नाउ रीड द क्वेश्चन, एंड सिंपल क्वेश्चन है, won't take much time. शिखा बताओ क्या होगा इसका आंसर.
Sir, option D.
Shika is saying option D, and that's a correct answer. Yes, meiotic division is all you need. You take karyotic meiosis and you'll find out all the megaspores have been formed. Yeah. Then, in angiosperm, the functional megaspore develops into. Tanish, बता दो क्या होगा इसका आंसर.
So embryo sac.
Right. So yes, your functional megaspore is going to develop into your embryo sac. Right. Very true. बिल्कुल सही. ठीक है. अच्छा, अब मेरा सवाल है आप लोगों से, तनिष, ऑल ऑफ यू, पोलन सैक में मुझे कौन से सेल्स देखने के लिए मिलते हैं? व्हाट काइंड ऑफ सेल आई गेट टू सी इन द पोलन सैक्स?
New cells.
शिखा, यू हैव टू रिपीट योरसेल्फ, कांट हियर यू, जोर से.
New
Sir,
cells.
I see.
इट इज एंथर के अंदर होता है बेटा पोलन सैक, उसके अंदर सेल्स कौन से होते हैं? व्हाट आर द सेल्स आई गेट टू सी इन द पोलन सैक्स?
Sporogenous tissue होता है.
Pollen.
जेनस.
Pollen mother cell.
Now, initially, you have simply going to tell them porogenous tissue or sporogenous cells. Whatever you can say is going to be a correct answer. Then what will happen? Some of the cells in the sporogenous tissue will turn into the microspore mother cell. So, ना आपके ये आपको मिलेंगे देखने के लिए, and then इसमें से आपके कुछ बनते हैं into your microspore mother cell. Okay, चलो आगे देखते हैं. मेगास्पोरेंजियम इज इक्विवेलेंट टू. 2013 का क्वेश्चन है, ये बहुत ही आसान क्वेश्चन है. टेलस मिसबा, क्या होगा इसका आंसर?
Option B.
ऑप्शन बी या डी? वापस क्या बोल रहे हो?
Option B.
बी. गायत्री, क्या लग रहा है?
Sir, option B, sir.
Yeah, it's going to be oval only. We have already done this multiple times. Male gametophyte in angiosperm produces. Again, you can answer this, or if you can answer this. Male gametophyte in angiosperm produces what? So male gametophyte in angiosperm. So male gametophyte is nothing but a pollen grain, right? You know that. So what are the things I'll get to see in that? Tell us, Misbah. बोलो बोलो.
Option D. No, sir,
Option.
option C.
हाँ, एक साइड पे ऑप्शन. ऑप्शन सी. शिखा, आपको क्या लग रहा है?
Sir, option D.
ऑप्शन डी. शिखा,
Option C, sir.
सोच के बोला करो, ऐसा मत करो. राइट. गायत्री.
Sir, option C, sir.
Yes, a male gametophyte is always going to have two sperm, that is my two male gametes, and one vegetative cell. So that is what a mature male gametophyte is going to have. Male gametophyte मतलब याद रखना, that is nothing but my pollen grain. Male gametophyte अगर कोई बोले, so that is nothing but my pollen grain. Right, it's my pollen grain. Now, filiform apparatus is a characteristic feature of. बताओ. Your गायत्री, बताओ क्या होगा इसका आंसर? Okay, Tanish, you tell us what will be the answer over here.
Targets.
Surrogate cells, right? Simple question. So these are the questions. Okay, function of filiform apparatus. I have just told you. Think and tell me. Function of filiform apparatus is going to be what over here. So take your time. रुक जाओ. अरे रुक जाओ. बताओ शिखा.
Sir, option D.
शिखा is saying option D. मिसबा, आपको क्या लग रहा है?
Option D.
Right, and I'm also assuming other students are also filling option D. Yes, guide the entry of the pollen tube. This is the one which is going to help in the guiding the entry of the pollen tube. That is what the filiform apparatus does. So please understand this thing. Filiform apparatus has a potential to release some chemical attractant. It has a potential to release some chemical attractant. So they are known to release some chemical attractant. And they actually help in the guarding the pollen tube towards the ovule, so that they can enter through the macropylent, right? The ovule of a angiosperm is technically equivalent to, again, we have done this question multiple times, and they have seemed to be answering this question multiple times, right? They have been seen to be asking this question multiple times. ये नीट में काफी बार आ चुका है, तो याद रखना ओव्यूल हमेशा आपका यहाँ पे रहेगा मेगास्पोरेंजियम. Hands down, the ovule is always going to be the megasporangium, right? Okay. Now, in majority of the angiosperm, which of this statement is going to be true? So think wisely. काफी जल्दी हैंड रेस कर दिया, तो सोच लो कौन सा स्टेटमेंट आपको यहाँ पे सही रहेगा इन कंपेयर टू द मेजॉरिटी ऑफ द एंजियोस्पर्म्स. Okay, Tanishq, tell us.
Sir, D part. A small central cell is present in embryo cell.
मिसबा,
Option C.
मिसबा वापस बोलना.
Option C.
Option C. All right. Shika.
Sir, option C.
Okay. So Tanishq, the thing is now, central cell is going to be large. Central cell, if you just see na, is going to be a large one. It is not going to be a small one, right? If you just observe over here, just come back. Let's quickly come back over here, right? आप यहाँ पे भी देख सकते हो, यहाँ पे छोड़ा हमारे पिक्चर में देखते हैं. If you just have a look over here, you will find the central cell is a large one. It's not small. Okay. So याद रखना central cell आपका large रहेगा, small नहीं रहेगा, बल्कि it's a very large cell in the embryo sac, right? So that's why that statement won't be correct there. ठीक है?
Okay, sir.
तो वो स्टेटमेंट आपको यहाँ पे करेक्ट नहीं आएगा. एंड इफ यू जस्ट सी द अदर स्टेटमेंट्स, नाउ यू कैन सी द अदर स्टेटमेंट. सो ऑप्शन सी यहाँ पे क्या कह रहा है? व्हाट द ऑप्शन सी इज टेलिंग अस ओवर हियर, reduction division occurs in the megaspore mother cell. That means it undergoes the meiosis. Reduction division basically means what? It's a meiosis. So that's a true thing, right? So that is why option C becomes a much more appropriate answer. It's a correct answer compared to the remaining three. Remaining three are going to be incorrect. See, agar has a filiform apparatus. That's wrong. It is something you find in synergid cells, right? There are numerous antibodal cells. That is also wrong. You only have 3 antibodal cells. So that is why option C over here becomes my correct answer. Okay. Which is the most common type of embryo sac in the angiosperms? Again, it's a logical question है, कर सकते हो आप लोग, कोई यहाँ पे difficult question है नहीं है. So take your time and then raise your hand. Which is the most common type of embryo sac in the angiosperm? Okay, now tell us, Shikha, क्या होगा इसका आंसर?
Sir, option B.
Okay. Gayatri, tell us.
Sir, option B, sir.
Tanishq.
Sir, is option number B.
Right. And I'm assuming the Misbah must be also having this option B over here. Yes, it is also going to be monosporic. Monosporic with a 3 sequential mitotic division. You find 3 times mitosis will take place over there with no cytokinesis. So that is why you will find option B over here becomes my correct answer. बिल्कुल correct answer मेरे यहाँ पे option B मेरे यहाँ पे बन जाता है. Okay. Functional megaspore in the angiosperm develops into, ये तो वैसे ही बोल दो अनम्यूट करके आप लोग, क्या बनता है ये? It is always going to be forming my embryos. ठीक है? Functional megaspore will always turn into the embryo sac, right? And it will undergo 3 times mitosis and will form giving the embryo sac. Okay. These are some more questions. So now question number 1 over here. During megasporogenesis, meiosis occurs in. During megasporogenesis, your meiosis will take place where? So take your time and then tell me. You don't have to take much time. Question number 1 का आंसर यहाँ पे क्या होगा बताइए मिसबा?
Option A.
Right. Option is going to be correct answer. Megaspore mother cell. Very true. Starting from the innermost part, correct sequence of the parts of inside the ovule. So you have to start from inside towards the outside. Right. Now this can be slightly difficult, but I think you can still do this. ये थोड़ा hodgepodge हो गया ना यहाँ पे आपका, तभी try करो. अंदर से बाहर के side पे. Go from inside to outside. What do you think? All right. Tanishq has raised his hand. Okay, tell us Tanishq, क्या लग रहा है आपको यहाँ पे?
The C part.
B part.
नहीं sir, C.
C part. Okay. Any other person?
Sir, option A.
See, I do understand यहाँ पे थोड़ा सा आपका okay, just wait a minute. Let me plug in the charger or else will disconnect. सब गायब हो जाएगा. देखो. अच्छा देखो. सबसे अंदर अगर देखा जाए तो अगर अंदर से बाहर हम देखें, if we see from the inside to outside, if we just see from the inside to outside, then I tell you why C cannot be the one. Because embryo sac, okay, nucellus, okay, integument, okay, but then they have the egg cell. That should be in the center, na? So that is why it won't be there. Right. Here it is getting mismatched, so that's why it might become slightly difficult to understand. Now, you have over here, you have an egg, you have a nucellus, you have an embryo sac, and then you have an integument. So this looks again like much more appropriate, but again, the embryo sac should have been before that the nucellus, and then you should have a nucellus over here. So that is why it will also not be answered in this, right? Then over here you will get to see you have an egg, you have an embryo sac, you have a nucellus, and then you have an integument. So this B over here becomes my appropriate answer. I can see you cannot very clearly understand the statement. So, okay. Okay, now here is the question. Female gametophyte of angiosperm is represented by. Female gametophyte of angiosperm is represented by. If you cannot read the question, I am reading for you. Female gametophyte of angiosperm is represented by A, B, C, D. Batao, Misbah.
Option C.
Yes. Female gametophyte is nothing but my embryo sac. Yes, that is what you will get to see. Okay. Now I have few questions. So this is what we have done. So in the today's class, we have seen your female gametophyte formation. We'll have to do few more things right now. Wait. But what we have seen by far, we have seen your carpels. So let's have a quick recap. अब आप लोग मेरी हेल्प करोगे recap करने में. You are going to help me out in this. We have talked about the carpels, right? Now, if you have like multiple carpels, then you call them a multicarpellary. If you have a single carpel, you call them like a unicarpellary, right? Or monocarpellary. Okay. Now tell me if I have 5 carpels, right? If I have 5 carpels, 5 pistil, and they're all fused. If they're all going to be fused, then उसके लिए क्या term use करेंगे हम लोग?
Syncarpus.
Very good. Right. The term is going to be my syncarpus. If they are going to be fused, then I'll use it like a syncarpus. हाँ जी तनिष्क बताइए.
यही बोलना था सिंकार.
अच्छा, okay, right. That's going to be called. अब तो unmute करके वैसे पूछ सकता हूँ. That's going to be a syncarpus, right? Apocarpus is going to be the one in which you will find all the pistils are going to be free over there. That is the apocarpus condition, right? Now you tell us that junction between funicle and ovule is going to be called as what?
Hilum.
Right.
Hilum.
That is called as my hilum. Very good. Okay. ठीक है. ये तो बड़ा आसान सवाल हो गया. कुछ difficult पूछते हैं. Now you tell me, if I want to form, now understand the question very clearly. If I want to form 100 eggs, to form 100 egg cell, how many times I have to carry out the meiosis? Tell me that. Form 100 egg cell, how many meiosis I have to carry out? How many? हाँ जी तनिष बताइए.
Sir, 100 meiosis.
100 meiosis. और कौन कौन एग्री कर रहा है 100 के साथ? सब लोग एग्री कर रहे हैं?
Yes, sir.
Yes, sir.
अच्छा, so you are saying the answer is going to be 100 meiosis. तनिष्क बता दो 100 meiosis के साथ हम कैसे बना सकते हैं हमारे 100 egg cell. So now you have to tell me that. Start. So now I'll give you the initial integrant. That is going to be your megaspore mother cell. So you have a MMC, which is a diploid cell. Now tell us Tanish what will happen. बताओ यहाँ से क्या होगा.
So meiosis 1 and meiosis 2 will take place successively, and they will form 4 megaspores. One meiosis will form 4 megaspore, in which 3 megaspore will degenerate, and only one will be left as functional megaspore. And in 100 meiosis, there will be 100 functional megaspore left. So each functional megaspore will develop into a embryo sac. So
Very
100 megaspore can convert into
बिल्कुल
100.
बहुत बढ़िया, right? That's true. So that is how you will get to see that you will have a single megaspore, and a single megaspore will give rise to your single embryo sac, and the single embryo sac, or every embryo sac has only one egg cell. So that is why you need to carry out 100 time meiosis, क्योंकि ये तीन तो जाएंगे, ये तीन तो आपके डिजेनरेटिव आपके यहाँ पे हो रहे हैं, तो एक मेगास्पोर आपके पास रहेगा, one megaspore is going to remain with you, and that megaspore is the one which will have the embryo sac, which will be having a single leg. It will have number of things, but egg to यहाँ पे you will have only one egg only over here. That is why yes, 100 meiosis for 100 egg. Now if I ask you, the question is now to form 100 pollen grains. To form 100 pollen grains, how many times I have to carry out meiosis? I have to make 100 pollen. Misbah, Shikha, Mohit, Aisha, if Aisha is here, no, Aisha is not here. Gayatri and Tanish, to form 100 pollen grains, how many times I have to carry out meiosis?
So 25.
You are telling me 25 times.
Yes, sir.
So Tanish is telling us 25 times I have to carry out the meiosis. And Misbah, आपको क्या लग रहा है मिसबा एंड शिखा?
Sir,
25. Shikha, what do you think?
Sir, 25 times.
Shikha, now you have to explain how that 25 is coming.
सर एक बार में तो मियोसिस करके 4 बन रहे हैं हैप्लॉइड, तो फिर 25 करेंगे तो 100 हो जाएगा.
Very good, right. So now you have a microspore mother cell, you have MMC microspore mother cell, and this cell is my like diploid cell, right? So this diploid cell will undergo meiosis and I will get 4 microspores. अब यहाँ पे ऐसा नहीं होता कि 3 degenerate हो रहे हैं. You will have all 4 of them are going to survive. Right? So these are my 4 macrospores. Right? So these are going to be your. You will find such questions. You will find such questions coming in the exam. So you'll have a 4 macrospores. Okay. So yaad rakhna, yes, as the Shikha told us that after one meiosis, right? You will after one meiosis, you will get around like 4 pollen grains. Okay. So if you need to have 100 pollen grains, then how many times you have to carry out meiosis? You can also do the cross multiplication and we'll get to have 25 times meiosis becomes the correct answer. Gayatri, समझ आ रहा है? Gayatri and all the other students over here. Is this clear to all of you? Gayatri, आवाज आ रही है नहीं आ रही? Gayatri. Gayatri is not there in the class. So that is why you will find 25 times meiosis done, so that you will get to have your 100 pollen grains will be formed. Okay. Now my next question is, I have one more question now before getting to this topic. My other question here is going to be, you tell me if the antibodal cell, now understand the question very clearly. You have an embryo sac, and embryo sac will have an antibodal cells, right? How many number of antibodal cells you have generally in the embryo sac?
3.
3 होते हैं आपके यहाँ पे embryo sac के अंदर, आपके 3 antibodal cell आपके यहाँ पे होते हैं. When you count the number of chromosomes, so they are all going to be, you know that they are going to be haploid over there, right? You know that they are going to be haploid. You tell me, when you count the number of chromosome in them, you realize कि इसके अंदर number of chromosome है, they are going to be, let's say, 24. So single cell has 24 chromosome. Single antibodal cell has 24 chromosome. So you tell me how many chromosomes will be there in the leaf of the plant. The leaf of plant will have how many chromosomes? इसके अंदर क्रोमोसोम्स का नंबर कितना होगा, या फिर मैं इसको थोड़ा चेंज कहता हूँ, इजी बना देता हूँ. लीफ में कितने क्रोमोसोम होंगे, या फिर मुझे ये बता दो, और यू कैन टेल मी हाउ मेनी क्रोमोसोम्स आई विल गेट टू सी इन द nucellus. How many chromosomes? Because the number is going to remain the same in both of them. That's why. So now tell me, think and tell me over here that if you have individual cell and if you see the one of the cell, you see they have 24 chromosomes and this is haploid cell. So now tell me, a leaf of a plant cell will have how many chromosomes?
116.
बेटा वापस बोलना.
Sir, 168.
160 तो ज्यादा हो गया बेटा.
So 24 ही रहेंगे.
24 ही रहेंगे लीफ ऑफ द प्लांट में. न्यूक्लियस में भी 24 रहेंगे. दोनों का आंसर वैसे जो भी निकालोगे तो सेम ही आएगा. न्यूक्लियस या फिर मैं थोड़ा और इसको वो करता हूँ अगर कंफ्यूज हो रहे हो तो. इंटीग्यूमेंट भी यहाँ पड़ा. न्यू इंटीग्यूमेंट के सेल. इंटीग्यूमेंट्स. एनीवन एल्स? शिखा क्या लग रहा है? What do you think, Misbah? Misbah, what do you think? What can be the answer over here? I hope you have understood the question. Individual cell is there. You see one of the cell, you found that it has 24 chromosomes and it's a haploid cell. So now I'm asking you that what will be the number of chromosomes will be there in the leaf of the plant, nucellus or integument? How many chromosomes can be seen?
48.
Someone is telling me 48. Who are that someone? That someone is Misbah या शिखा. कौन है ये? गायत्री.
No, sir.
Misbah. Shikha. Misbah. Okay. Misbah, how 48?
Sir, plant cell diploid.
Right. Very true. Yes. देखो, leaf of the plant cell, leaf जो होगा plant cell, the leaf which will be there on the plant, always going to be diploid. Right. Nucellus are also going to be diploid. Even the integuments are going to be your diploid only. So if you have a haploid cell and haploid cell has 24 chromosomes, I hope you are understanding. If the haploid cell has the number of chromosomes in a haploid cell are going to be 24. So simple si baat hai, if I give you a diploid cell, then the number of chromosomes are going to become 48. Double ho jayega na uske andar. So leaf of the plant is going to be diploid, nucleus is going to be diploid, and also my integument is going to be diploid. So that is why 48 will become a correct answer over here. Is this clear to you, Tanishk and all the other girls over here? समझ आया?
Yes, sir.
ठीक है. तनिष्क इसको लेके कोई कंफ्यूजन तो नहीं है ना?
No, sir.
All right. Cool. ठीक है. So that is what you will get to see. So please have a habit of also seeing such kind of question as well. So अगर मैं यहाँ पे लिखता ना root tip, if I've asked you that individual cell are going to have 24 chromosomes and what will be the number of chromosomes in a root tip, then also you could have said ki nah, it is going to be my 48 only, because if they are going to be 24, then that's going to be 48. Now let's do one thing. Let me ask you one more question like this. Just one more question over here. Let me frame a question over here. See, now tell me, in the embryo sac, you have synergate cell. How many synergate cell you have?
2.
Right. Right. So you always have 2 synergate cells, right? You will have 2 synergate cells, right? And you know that both of them are going to be haploid. Now, when you observe, when you count the number of chromosomes in them, the number of chromosomes in them are going to be 12. The number of chromosomes in them are going to be 12. Now you tell me how many number of chromosomes I'll be finding in my microspore. How many chromosomes I'll be finding in my microspore, which has been abhi bana hoga mera from the microspore mother cell se. So if a new microspore has been formed, how many chromosomes it will be happening? It will be having.
Only.
Again, आप लोग चुप हो गए. Microspore, you know na how it has been formed. It is going to be again haploid structure.
12 only.
You have a diploid microspore mother cell. It will undergo meiosis and give rise to the microspore. And microspore are also going to be haploid. If a microspore is going to be haploid, you have a haploid which is going to be 12. So it is also going to be 12 over here. ठीक है? Confused नहीं होना है. ये बड़े ही आसान सवाल है. And you will find such question and you should be able to answer this. कर लोगे तुम लोग. बड़े simple question है. Okay? That is something you have to know. All right. So that is something we have done in today's class. So I will be sharing with you an assignment which will again have around 12 to 15 questions based on this only, right? This particular topic. And you might find a couple of questions from pollen grain as well. So you can have a look at them. Now, before we wrap up the class, you can also do this question as well. In the anther wall, what do you think, which is the statement which is going to be much more appropriate? Take your time. Which statement?
The part.
C. All right. C is going to be a correct answer. Yes. Middle layer lies between the endothelium and the tibitum is going to be a correct answer. Very true. Okay. I'll just tell you one more thing here. A double layer wall of pollen grain is called as my sporoderm. This, you know that your pollen grain will have a two walls. No? You have your intine and exine, right? You have an intine and you have your exine as well. So intine and exine, these two walls are collectively have a single term. This inner and outer wall will have a single term, and that single term is going to be my sporoderm. So this inner and double layer wall of your this pollen grain is called as my sporoderm. So that should be clear to all of you, right? So this is what we have done. We are done with the female gametophyte. When we'll meet next time? So first of all, you have to read this thing thoroughly. Just go to the NCERT once, and I'll share you the questions. Please solve those questions, right? When we meet again, we'll be starting with the pollination, and we'll try to complete that in a single lecture, which will eventually happen, right? So that will be the target for the next class. And this generally, this lecture doesn't take more than 6 to 7 hours, right? Collectively to get completed. That is what we'll try to do. Okay, so that is something we have done in today's class. So yeah, that's it. Now, if you have any questions or is there something you haven't understood, agar kuch samajh nahi aaya ho, then also you can ask a question. We have roughly done around like 12 to 15 questions. You also get to have an idea that what kind of question they're going to ask you in the NEET exam. So we have also done that, right? And additional, I'll share some questions with you, which you can practice at your home just to have a better grip on the topic. Okay, so now tell me, just wait a minute. Yeah. Now tell me,